Hi everyone. In this video, I am going to explain you about the remaining topics of Monera. Staining of bacteria by gram staining technique. First of all, HC gram differentiated bacteria based on staining. In the first step of this method, bacteria are stained with crystal violet or gentian violet and the Ki solution. After staining, bacteria are washed with acetone or ethyl alcohol. After washing, some bacteria retain the stain and some bacteria are decolorized. Bacteria which retain stain, violet or purple, are called gram positive, and bacteria which decolorizes are known as gram negative. Gram negative bacteria are counterstained by safranine. Nutrition in bacteria. Compared to many other organisms, bacteria as a group shows the most extensive metabolic diversity. Most of the bacteria are heterotrophic, but some are autotrophic. On the basis of nutrition, bacteria are classified into three categories: autotrophs, heterotrophs, and symbionts. Autotrophs. These bacteria use light or chemical energy for their own food synthesis. On the basis of source of energy, autotrophs are following two types. First one, photosynthetic autotrophs. These bacteria uses light energy for food synthesis. Photosynthetic pigments are present in cytoplasm for photosynthesis. In these bacteria, photosynthesis is non-oxygenic. Some photosynthetic bacteria are purple sulfur bacteria, example chromatium; green sulfur bacteria, example chlorobium and thiothrix; purple non-sulfur bacteria, example rhodospirillum and rhodopseudomonas. Chemosynthetic autotrophs. They use chemical energy instead of light energy for food synthesis. Chemical energy is obtained from oxidation of chemical compounds. They may be inorganic or organic. Example, nitrifying bacteria. They oxidize nitrogenous compounds and obtained energy. See the formula: ammonia in the presence of nitrosomonas or nitrococcus gives NO2. In the presence of nitrobacter, it gives NO3. Note: chemisynthetic bacteria play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophs. Most of the bacteria are heterotrophic. That means they cannot manufacture their own food. The majority of heterotrophic bacteria are important decomposers. Heterotrophic bacteria are useful in making curd from milk, production of antibiotics, fixing nitrogen in leguminaceae plants. Some are pathogens to human beings, animals, and plants. They receive their own food from dead organic matter or living organism. Saprophytic bacteria. These bacteria obtain food from dead and decaying organic matter. These are of two types. Number 1, obligate saprophytic bacteria. These bacteria obtain food only from dead organic matter. These are completely saprophytes. Example, Bacillus vulgaricus, Clostridium botulinum. Facultative parasite. These are normally saprophytic bacteria in nature, but in the absence of dead organic matter, they can become parasitic. Example, Pseudomonas and Staphylococcus. Parasitic bacteria. They obtain their food from living organism. Parasitic bacteria also two types. Number one, obligate parasite. They always remain parasitic. Example, Mycobacterium leprae. Facultative saprotrophic. They are normally parasitic in nature, but in the absence of living host, they may become saprotrophs. Example, Mycobacterium tuberculosis. Respiration. Based on respiration, bacteria were classified into following types. Very first one, obligate aerobic bacteria. These are completely aerobic and die in the absence of oxygen. Example, Azotobacter. Facultative anaerobic. These are normally aerobic but can survive in the absence of oxygen also. Example, Acetobacter aceti, Clostridium tetani. Coming to anaerobic bacteria, obligate anaerobic. These are completely anaerobic bacteria and do not have capacity of aerobic respiration. Example, Clostridium botulinum. Facultative aerobic. These are normally anaerobic but also have capacity of aerobic respiration. Example, Lactobacillus. Reproduction. Bacteria reproduces by two methods. Asexual reproduction. Very first one, binary fission. This is the most common method of bacterial reproduction. Under favorable conditions, first of all DNA replication takes place in bacterial cell. Then, bacterial cell divides into two daughter cells due to the formation of transverse septum in the center of the cell. Each daughter cell grows into a new bacterium. Under favorable conditions, the cells of bacteria divides by amitosis, which is faster than mitosis and meiosis. By endospore formation. Endospore formation occurs under unfavorable conditions in bacteria. Endospore formation is highly resistant structure. It is resistant to temperature, radiations, antibiotics and chemicals. Endospore is highly resistant structure due to presence of calcium hypocolinite in cortex. Endospore formation is seen in mostly bacillus type of bacteria. Second one, genetic recombination. Genetic recombination includes three methods: transformation, transduction, and conjugation. Transformation. First explained by the scientist Frederick Griffith in Streptococcus pneumoniae. Uptaking of naked DNA from surrounding environment and express the same genetic information in recipient cell is called transformation. Second one, transduction. First explained by the scientists Lederberg and Ginder in Salmonella. Transferring of genetic material from one bacteria to another bacteria through viruses or bacteriophages called transduction. Conjugation occurred between F plus and F minus. It is a sort of sexual reproduction. Conjugation was first discovered in the year 1946 by the scientists Lederberg and Tatum in E. coli. First of all, donor F plus is attached to recipient F minus with the help of sex pili. Sex pili functions as conjugation tube. The F factor, F plasmid, now replicates and the R replica moves to F minus through conjugation tube. Both the cells are then separated due to transfer of F factor. F minus bacteria now becomes into F plus bacteria. Economic importance of bacteria. Very first one, harmful activities. Very first one, diseases in human beings. Tuberculosis, TB, caused by Mycobacterium tuberculosis. Leprosy caused by Mycobacterium leprae. Tetanus caused by Clostridium tetani. Typhoid caused by Salmonella typhi. Cholera caused by Vibrio cholerae. Diseases in animals. Anthrax caused by Bacillus anthracis. Blackleg caused by Clostridium chuevi. Diseases in plants. Citrus canker caused by Xanthomonas citri. Crown gall diseases in many plants, especially in apple, caused by Agrobacterium tumefaciens. Blackleg and soft rot of potato caused by Yervinia carnivora arthroseptica. Bacterial leaf blight of rice caused by Xanthomonas varice. Denitrification, denitrifying bacteria. Some bacteria convert soil nitrates into nitrites and nitrogen. These bacteria reduces the fertility of soil. Example, Thiobacillus denitrificans and Pseudomonas denitrificans. Food poisoning. Botulism. Clostridium botulinum. It is most lethal type of food poisoning, causes paralysis of both smooth and striped muscles, resulting in immediate death. Water pollution. Several bacterial forms causes water pollution. These bacteria spoil the water. Example, Vibrio cholerae, Salmonella typhi. Biological weapons. Some bacteria are used as bioweapons, such as anthrax causing bacteria, botulism, and cholera causing bacteria. Useful activities. Very first one, ammonification. Some bacteria convert protein into ammonia, present in decaying plants and animals. Example, Bacillus vulgaricus. Nitrification. Bacteria convert ammonia into nitrite and nitrate. Example, Nitrococcus nitrosomonas. Nitrogen fixing. Some bacteria convert atmospheric nitrogen into nitrogenous compounds. Dairy products. Streptococcus lactis, lactobacillus helps in curdling of milk. Especially lactobacillus or lab or lactic acid bacteria increases vitamin B12 and checking disease causing microbes in stomach. Antibiotics. The term antibiotic was given by S. A. Baxman. First discovered antibiotic was penicillin. It was obtained from fungi Penicillium notatum. First discovered antibiotic from bacteria was streptomycin. Many antibiotic medicines are obtained from the bacteria. Some substances produced by microorganisms which inhibit the growth of other microorganisms are called antibiotic substances. Industries. Vinegar formation or acetic acid formation by Acetobacter aceti. Rating of fibers. Separation of fibers from plants. Example, Clostridium butyric acid bacteria. Flavoring or curing of tea leaves and tobacco leaves. Example, Bacillus megatherium and Micrococcus condensans. Production of vitamins. Clostridium botulinum produces vitamin B2 and butyric acid. Propionibacterium and Bacillus megatherium produces vitamin B12. E. coli produces vitamin E and vitamin K. E. coli bacteria found in alimentary canal of human beings. Purity of Ganga water. In Gangatic water, a bacteria, Delovibrio bacteriovirus, is found. They kill the other water polluting bacteria. Pollution indicating bacteria. Water in which E. coli bacteria are present known as polluted water. Quality of water depends on number of E. coli. If E. coli are very much in number of in the water, will be highly polluted. So the E. coli is known as pollution indicating bacteria. Bacteria for genetic engineering. Example, E. coli and agrobacterium.